Okay, next we will discuss types of immunofluorescence, and first of all, type 1. This is direct immunofluorescence. When we are using direct immunofluorescence, it means there is one-step staining using a single and labeled primary antibody. And when we say labeled primary antibody, it means it is conjugated directly with some fluorophore. Labeled primary antibody is able to bind with specific epitope of the target antigens in the given sample. So this targets one particular site of the antigen. It's not targeting the whole of the antigen, one particular part of the antigen, which is named as epitope, that is stained with this labeled primary antibody. Antigen is visualized via attached fluorophore by fluorescent microscopy. So this attached fluorophore, that is actually the factor which is responsible for visibility of that particular antigen. The fluorophore emits a light of specific wavelength on excitation when it is exposed to the specific radiation. Here we can see the principle of direct immunofluorescence. Here we can see the tissue at the bottom, and this tissue, of course, express different types of antigens, different types of proteins. These proteins, they are targeted with the help of using antibodies. Here we can see blue color primary antibodies. These specifically target those proteins or those antigens in the tissues. And these primary antibodies, these are labeled with fluorophore, which is here fluorescein, which is shown in green color. So antigens present in the tissues, they are stained with these primary labeled antibodies, which are directly labeled with some fluorophore. And here secondary antibodies, they are not used. So primary labeled antibodies, they are used here. So direct attachment of the fluorophore to the antibody reduces the number of steps in the staining, which saves times and reduces non-specific background signals as well. So if we compare this direct immunofluorescence with indirect fluorescence, which we will discuss later on, it is more beneficial. The reason is that it is one-step staining. Only in one step we are able to see the cells, and similarly there is background signals. There is reduced background signals. At the same time, direct immunofluorescence is quite fast, as there is less washing and less incubation steps as compared with indirect immunofluorescence. It limits the possibility of antibody cross-reactivity as it is directly labeled. So there is no problem, or there is less problem of cross-reactivity in direct immunofluorescence. And similarly, as the number of fluorescent molecule is limited, it is substantially less sensitive than indirect staining. So this is disadvantage or limitation of direct immunofluorescence, that this is less sensitive as compared with indirect staining.